Welcome to another installment of the Library Seminar Series, Knauss Fellows 2020. I'm Lisa Clark, Reference Librarian at the NOAA Central Library. Today's library seminar features 2020 Knauss Fellow Grace Roskar, who will discuss elasmobranch community dynamics in Florida's Indian River Lagoon. Mike Aquafita, a 2020 Knauss Fellow with the NOAA Ocean Acidification Program, will introduce our speaker. Before the presentation begins, here are a few logistical tips. If you're having trouble with the audio or visual components of GoToWebinar, please log out and rejoin us. This usually resolves technical issues. This presentation is being recorded and will be available on the NOAA Central Library YouTube channel, as well as the Library Seminars website by later this afternoon. We will also be accepting questions throughout the webinar, which the speaker will address at the end of her presentation. Please feel free to type your questions in the question chat box on the right side of your screen at any time. So with that, I thank you for attending this library seminar and turn this presentation over to Mike.
Thank you, Lisa. It's my pleasure to introduce Grace Roskar, a 2020 Knauss Fellow in NOAA Fisheries Office of Science and Technology, supporting the Marine Ecosystems Division as a habitat science specialist. Grace holds a Bachelor's of Science from the University of Miami and a Master's from Florida Atlantic University's Harbor Branch Oceanographic Institute. Her graduate research focused on estuarine elasmobranch ecology in Florida, as well as evaluating fishing gear performance in a fishery-independent survey. With that, I'll pass it over to our speaker, Grace. Grace, you can take it away.
Hi, everyone. Thanks so much for attending my presentation today. I'm going to be speaking about my master's research that I did at Harbor Branch with the Fisheries Ecology and Conservation Lab, which was under the advisement of Dr. Matt Ajamian. Sorry, technical difficulty. Okay. So my research was focused in the southern part of the Indian River Lagoon, or the IRL for short. I'll be referring to it as the IRL throughout this presentation. The IRL spans 250 kilometers and comprises approximately 40% of the state's east coast. And it's actually comprised of 3 different bodies of water: the Mosquito Lagoon, the Banana River, and the Indian River Lagoon proper. 5 inlets across the lagoon provide access to the ocean, while freshwater enters the IRL through rainfall, surface water runoff, inflows from tributaries, and canals, some of which are depicted by these red boxes. So for example, the C44 canal at the bottom is connected to Lake Okeechobee and is used for flood control. So they'll release fresh water from the lake. It'll flow through the canal into the lagoon. And this canal, along with these 2 other canals at the bottom, supply a lot of fresh water to the St. Lucie River, which feeds into the IRL, which I'm highlighting with my mouse if you can see it down there. And then there are various other canals throughout the lagoon as well. So the IRL is one of 28 estuaries designated as an estuary of national significance by the Environmental Protection Agency's National Estuary Program, which aims to conserve and manage important estuaries in the US. And the IRL has long been said to be extremely biologically diverse, home to many different species of organisms, some of which you can see here. And so my research focused on the elasmobranchs, or the sharks and rays that inhabit the lagoon. So why do we care about elasmobranchs? Many of you listening are probably aware of both the economic and ecological importance of elasmobranchs, and also how some certain aspects of their life histories, such as their long lifespans and low fecundity, lend low population growth rates and make them particularly susceptible to threats such as overfishing and habitat loss. However, these animals are important, as many of these species hold high trophic positions as apex or mesopredators, and they help maintain balance within complex marine food webs. Additionally, many ray species consume crustaceans or mollusks, and they can therefore act as ecosystem engineers. So they'll create pits in the sediments while feeding, and this can create habitats for benthic organisms and cause bioturbation of the sediments. And then additionally, elasmobranchs are important economically. They contribute to ecotourism industries, and they also often act as a food source in many coastal countries. And so then why do we care about estuaries for elasmobranchs? Well, estuaries like the IRL are important habitats for many different species. Some elasmobranchs use estuaries to give birth. Some utilize them as nurseries in their early life stages, which is thought to help increase survival due to increased prey abundance and decreased predation or competition risks. And then many species use estuaries as foraging grounds as well. So then why focus on elasmobranchs in the IRL specifically? Despite the significance of the IRL, little is actually known about the elasmobranchs that inhabit this estuary. So if we were to take a look at the timeline of the last 50-ish years in the lagoon, there was a seminal paper published by Grant Gilmore on the fish diversity in the lagoon back in the 70s, and he documented 13 species of elasmobranchs caught between 1971 to 1975. Snelson and Williams collected 10 species from 75 to 78. However, their study didn't even cover the southern portion of this large lagoon. And then back in the 90s, the Florida Fish and Wildlife Conservation Commission started their fisheries-independent monitoring program to survey the overall fish community. And a paper that was published in 1995 that did list a few elasmobranch species, but it didn't include any analysis of that information, and their monitoring was only based, again, in the northern part of the lagoon. And then there's this large gap in published studies until 2011, when Toby Curtis and his colleagues published an analysis of 30 years of bull shark data to address their distribution and habitat use. And he was able to determine that the northern IRL functioned as a nursery for bull sharks, but he didn't have enough data to make the same determination for the southern IRL. And so also in this large gap between studies, heavy fishing and a lack of regulations started depleting many shark species in the Atlantic. And as a result, in 1993, the first fishery management plan for sharks in the Atlantic was introduced to try to curtail overfishing of shark stocks. Also during this gap of time, the IRL was and still is experiencing significant population growth in the area. And this is resulting in land use changes and coastal development, which have all had an impact on the health and water quality of the lagoon. And so this resulted in the first comprehensive conservation and management plan for the lagoon back in 1996. So we can see that from these management measures, there was an impetus to start to manage and monitor these resources. However, today we're still experiencing the effects of anthropogenic actions in the lagoon, and we see it through events like harmful algal blooms that have been occurring in recent years as a result of freshwater discharges and eutrophication. So as these changes continue to impact the estuary, we really need baseline information on the elasmobranch community in order to understand how they may still be affected in the future. So this brings me to my project. What little we know about elasmobranchs in the IRL is generally pretty much decades old, as you can see here, and especially lacking in the southern portion of the lagoon. So the goal of my research was to fix that. So estuaries like the IRL are dynamic and complex habitats. Thus, there are often many different factors that are at play influencing species assemblages. So the objectives of my research were to use a fishery-independent survey to characterize the composition and distribution of elasmobranchs in the southern IRL, examine how this varies by time and space, and also investigate how abiotic parameters may shape these assemblage patterns. So with this, we hope to gain an understanding of how different species utilize the IRL throughout their life histories and better understand the influences of abiotic parameters on that elasmobranch distribution. An additional part of my research was determining the performance and effectiveness of the different fishing gears we use to conduct the survey. But in the interest of time today, I won't be presenting on that part, but I'd be happy to take questions later on it. So I'll be focusing on these first three objectives today. So to accomplish these objectives, we implemented the fishery-independent survey. We started, our survey area was these 5 regions you can see here. So from the Sebastian Inlet in the north, all the way down to the St. Lucie River and the St. Lucie Inlet in the south. We broke up the IRL into these 5 major regions, which you can see on the map: Sebastian, Vero Beach, Fort Pierce, Jensen, and St. Lucie River. And I'll be referring to these regions a lot during this presentation. And then for logistical purposes, we broke down each major region into 2 subregions, which are noted by the A's and the B's on the map here. Each subregion was sampled 1 day per quarter or season, and the breakdown of our seasons are listed here. And the survey started in July 2016, and the data was analyzed until June 2018 to give a full 2-year data set. 2 types of fishing gear were used in the survey, the bottom longline and the gill net. So the bottom longline follows a standardized gear design developed by NOAA's COSPAN survey. COSPAN stands for the Cooperative Atlantic States Shark Pupping and Nursery Program. And this is a collaborative effort with federal, state, and university partners to survey and monitor shark habitats along the Atlantic coast to determine nursery areas and help refine essential fish habitat designations. So this gear comprised of a 300-meter long main line with 50 monofilament leaders with baited circle hooks that were clipped to the main line. 25 of these hooks were baited with Boston mackerel, and 25 hooks were baited with striped mullet. Mackerel is a commonly used bait in elasmobranch surveys, and then striped mullet is locally available and is also a local species that occurs in the lagoon. So the longline was set parallel to the shore and soaked for 30 minutes. And then we also used a gill net. So this gill net was comprised of 2 panels of mesh. Each panel was 100 meters long and 3 meters deep. So the whole total net was 200 meters long. One panel had mesh that was 15.2 centimeters or about 6 inches wide, and then the other panel was 20.3 or about 8 inches. The gill net was set about 15 minutes after the longline, around half a kilometer away. And the gill net was set in a line perpendicular to the shore and soaked for about 45 minutes. So once an elasmobranch was captured, we brought it on board. The species and the sex was recorded. Their weight was taken along with various length measurements. The ones I'll be talking about in this presentation are fork length for sharks, which is the length from the tip of the snout to the fork of the tail, and then disc width for rays, which is the length or distance between the 2 wingtips. Several samples were also taken, including a blood sample, a fin clip, which is like a tissue sample, and then microbiological swabs of the mouth and the cloaca. And then each animal also received an external numbered ID tag, which was provided by the COSPAN program, and then it was released back into the lagoon. So in addition to the data we collect on the animals in the survey, we also took environmental data. So for each longline and gill net set, we marked the coordinates, the tidal status at the time, the minimum and maximum depth over which the gear is set, and then I calculated the distance to the nearest inlet and the distance to the nearest freshwater source using GIS. And then we used a YSI to measure the typical environmental parameters at both the surface and bottom of the water column, such as temperature, salinity, and then we used a Secchi disk to measure water quality. So to jump right into the results, over the 2 years, we caught 630 individuals of 16 different species, which you can see listed here. Rays comprised about 61% of the catch, while sharks comprised 39%. Bull sharks and Atlantic stingrays were the most common species. You can see we caught 164 bull sharks and 133 Atlantic stingrays, and then other species ranged were a little less common. So this slide shows the regional presence or absence of each species. So the species are on the bottom x-axis, and then the regions run from north to south on the y-axis. The black bars show that the species was present in that region. So just to point out some highlights, each of the top 6 most commonly caught species were caught in all 5 regions of the lagoon, with the exception of the bonnethead shark, which was not caught in the St. Lucie River. Bull sharks were the actual only shark species caught in the St. Lucie River. And the lack of other shark species in this lower salinity region may be a result of those species' higher salinity preferences. So the St. Lucie River, as I mentioned, was connected to the freshwater canals, and so it often had very low salinity. So this slide is similar, but shows the seasonal occurrence of each species. It's kind of flipped. The species are now on the y-axis and the months and season are running along the bottom. So another highlight, Atlantic stingrays, blunt-nosed stingrays, and bull sharks were present every single month of the year, indicating that they're year-round residents of the lagoon. Meanwhile, other species were only present during certain times. So for example, sandbar and fine-fused sharks were only present in the first 5 months of the year. And this suggests that the species may use the IRL as an overwintering ground, and likely they move elsewhere during warmer months. And then sparse absences of other common species such as cownose rays, southern stingrays, and bonnetheads are probably due to lower sampling effort. Other studies on the thermal tolerances of these species don't indicate that they would likely leave the lagoon in these kind of patchy random months. So we think it's just because sampling days weren't always in the exact same month. So to test to see if there were significant differences in the species assemblages among regions and seasons, I used the permutational multivariate analysis of variance, or PermaNOVA analysis, for each gear type. So this slide shows the longline results of the PermaNOVA. So as you can see here, the longline region did have a significant effect on the composition of species caught, whereas season did not, nor did the interaction between region and season. So looking further into this, we can see that the species assemblages in Sebastian, Vero Beach, and Fort Pierce are similar, indicated by the letters that they share here. But Fort Pierce is also similar to Jensen Beach, and then Vero Beach and St. Lucie River are similar as well. And then this, the fact that there's no significant difference between the St. Lucie River and Vero Beach regions is likely due to the freshwater input in each of these regions, which I've mentioned a few times already, because of the canals that are present in Vero Beach, which you can see with these black triangles, as well as the canals in the St. Lucie River. So further analysis revealed that bull sharks were the top species contributing to these differences between regions. They were more common in St. Lucie River and the Vero Beach regions over Fort Pierce and Jensen and Sebastian. And this is likely due to the fact that bull sharks do have the ability to tolerate a wide range of salinities. It's been reported they can tolerate between 0 to 50 parts per thousand. So then moving on to the Gill Net results, the same analysis showed that only season had a significant effect on the species composition. And looking further into that, we could see that the species assemblage in the fall is statistically different from that of both spring and summer, whereas the winter assemblage isn't significantly different from any other season. So looking further into the actual species causing these differences, we saw that bull sharks, Atlantic stingrays, and blunt-nosed rays were the top species contributing to these differences. So let me just back up here real quick. Bull sharks and Atlantic stingrays were more common in the fall than both the spring and summer, whereas blunt-nosed rays were less common in the fall and more common in the spring and summer. You can also see here that the catch numbers between seasons did vary quite a bit. The higher abundance of bull sharks and Atlantic stingrays in the fall is likely attributed to the low salinities of that season due to precipitation during the beginning of the wet season, and also freshwater discharges from the canal due to higher precipitation levels. And then the higher abundance of blunt-nosed rays in the spring and summer over the fall is likely related to the gear selectivity for certain sizes. So blunt-nosed rays give birth in the IRL from May to June, which coincides with the spring sampling season. So it's possible that as neonates or newborns, the blunt-nosed stingrays are too small to be captured by the gill net in their first year of life through the fall and winter, but then they likely grow large enough that by the following spring, they can be caught in the gill net, which would explain the higher abundance of these rays in the spring and summer. So moving on to assessing how abiotic parameters affected species assemblages. I mentioned earlier that we measured these environmental variables each time we set the gear, but I also needed to first calculate the distance from each set that we caught in Elasmobranch to the nearest inlet, which is marked by the green stars here, and then also the distance to the nearest canal, which are marked by the blue triangles. So to do this, I use the cost distance function in GIS, which lets you measure the distance between 2 points, but it does take land into account. So this map is a result of using that tool in GIS, and what it's showing is that the areas in red or orange are closer to the inlets or the canals, whereas the areas that are yellow or green are farther away. And so each of these dots represents one of the longline or gill net sets. So you can see, if they're in red areas, they're closer to an inlet. And it actually, the tool does give a distance estimate. And so I use those numbers along with the measurements of all of these other environmental variables. And I used the relate test, which is a non-parametric version of the mantle test in the software program Primer. And so this test tests the null hypothesis of no correlation between the biological or catch data and the environmental data for each gear type. So as you can see here, the relate test produced a significant result. And so then to look further into it, I used the biota environment matching analysis, or the bio-ENV. And this test determines the greatest Spearman rank correlation values between the environmental variables and the elasmobranch catch. So basically seeing which environmental factors were driving this variation of the species assemblage with each gear type. So for the long line, we can see that water clarity and distance to freshwater source were the two factors that were driving the species assemblage the most. And these two environmental parameters are related, as water clarity generally does decrease with lower salinity. And then I used a principal components analysis, or PCA, just to show a visual of how the environmental conditions are overlaid with the species composition. And so looking at this, we can see a multi-species assemblage in clearer, higher salinity waters that are closer to the inlets, whereas a single species, which is bull sharks in the pink, is dominating the turbid, the less clear, fresher water that is farther from the inlets. So I did the same analysis again for the Gillnet. The relate test produced a significant result again. So I used that bio-ENV test, and this showed us that the combination of depth, salinity, and distance to an inlet were the abiotic factors driving this set of species the most. So depth and salinity are associated with proximity to the inlets in the IRL, because in the inlets there are dredged deeper channels, and there's higher salinity seawater that's entering the lagoon through inlets. It's also important to note that the longline did have higher correlation values than the Gillnet. And so while these correlation values are still low, considering that these species are highly mobile and somewhat rare, these low ranges of values are generally accepted in the literature. So the PCA for the Gillnet, again, shows a higher diversity of species in deeper, higher salinity waters, whereas bull sharks in the pink and Atlantic stingrays in the maroon start to dominate the catch more in shallower, lower salinity waters that are farther away from them. So now I'm going to switch gears a little bit from the environmental analyses to talk a little bit more in depth about some of the important species we caught in the survey. So these graphs show the frequency of which we caught certain sizes of some of our common species. So you have the length at the bottom, and then the frequency or number along the y-axis. And then these dashed lines show the length at sexual maturity for each sex. So blue for males and yellow for females. So important takeaways from this, we can see that no mature bull sharks were caught in the survey, whereas for many other species, we did catch both immature and mature individuals. This could be an artifact of gear bias, meaning that maybe our hooks were too small to catch the bigger sharks, or the net wasn't the right size, maybe the bigger sharks would break through. But we don't think this is generally true, because we did catch large specimens of other species. For example, in the Gill Net, we caught large nurse sharks. But what we think is happening here with the bull sharks is that it's more likely that bulls that are greater than about 160 to 108 centimeters are large enough to leave these nursery habitats and transition to adult habitats offshore or out of the lagoon. So this is really telling us that it's evident that some species use the lagoon throughout their life histories, others use it only on certain times, and we may be able to study these animals in the future to see the effects, especially the animals that do use the lagoon throughout their life. We can maybe start to look at the effects of long-term exposure to impacts such as reduced water quality or harmful algal blooms. So I also examined how size varied by space or region for some of the common species. So with these graphs, the region is abbreviated at the bottom, and then the length is along the y-axis. The graphs show the minimum, maximum, and mean fork length or disc width for these top 3 species. The dashed lines again show the length at sexual maturity. But really, I just want to point out a few key differences. So starting for bull sharks, the mean length of bull sharks was significantly different depending on the region. We caught the smallest bull sharks in Vero Beach and St. Lucie River, which are, again, the regions that generally had lower salinity waters. And so we think that the utilization of these lower salinity waters may be a predator avoidance strategy. Larger predators may not enter these areas. Or it could also be related to a physiological need to minimize the metabolic costs of osmoregulation for these smaller individuals. For Atlantic stingrays in the middle here, the mean size was highest in Vero Beach and St. Lucie River, which again are these areas that are farther from inlets and have more freshwater input. And so it's possible that in these areas, or actually in areas of higher salinity outside of these areas, other ray species such as larger blunt nose and southern stingrays are more prevalent than in these areas that have more freshwater influence. And so it might reduce the dominance of larger Atlantic stingrays, especially considering the diets of the 3 species are similar and therefore competition may be higher. So in these lower salinity areas in Vero Beach and St. Lucie River, the Atlantic stingray, because it can tolerate these lower salinities, it might be able to dominate the catch and dominate the area. So another notable catch during the survey was the capture of 2 smalltooth sawfish in the St. Lucie estuary. This is a critically endangered ray species, and it was especially exciting and important because one of the papers I mentioned by Snelson and Williams back in 1981 actually speculated that this species had been extirpated from the system, even though it was once so abundant that it was actually known as a nuisance to fishermen in the lagoon. Since then, there have also been numerous reports of sightings and captures of smalltooth sawfish of all different sizes by fishermen within the IRL. So we got lots of reports, other people got reports too. And so in addition to our 2 captures, this might indicate that the species is starting to recover in the lagoon. And yeah, it's really exciting. And I'll come back to this in a little bit. So overall, the survey did serve to fill the substantial data gap that surrounded the status of elasmobranchs in the IRL. This table shows the species caught in the previous studies that I mentioned compared to ours. And those highlighted in orange are ones that were not previously recorded, but that we did catch. So Atlantic sharpnose, blacknose, and fine-toothed sharks are smaller coastal species. And it's possible they weren't in the system before, or they just weren't encountered because of differences in fishing gears between our study and the previous study. But either way, it's still notable that we do have an increase in the record of elasmobranch diversity in the lagoon. Lemon sharks are the one species that eluded our survey, but have been documented in the lagoon. Honestly, we're really not quite sure why. We did do a few opportunistic night sampling events, and we did catch some large lemon sharks at night, but we never caught them in our survey, even though they've been reported and we've actually seen them, when we weren't doing the survey. So we know they're there. I'm not sure why we're not catching them. Don't know too much more about them in terms of the southern part of the lagoon. They there's more research in the northern part of the lagoon around Cape Canaveral of the species, but for us, it's still kind of a mystery. So in general, this survey was the first standardized elasmobranch specific survey, meaning targeting elasmobranchs in the southern IRL. It provided us with an overall picture of the community as well as additional valuable information for certain species. It's likely that the southern IRL serves as a nursery for bull sharks. We can't quite make that determination quite yet because there is certain criteria that accepted in the elasmobranch scientific community of what constitutes a nursery, and I can go into more detail later if there's questions about it. But it is likely a nursery, but some additional studies on using acoustic telemetry to track these individuals would be helpful to make the full determination that the southern IRL is a nursery. So additionally, as I mentioned, the capture of these 2 smalltooth sawfish, in addition to the other reports in the area, may indicate population recovery in the area, which is really exciting. And this survey, it really provided baseline knowledge for the start of what will be continued sustained monitoring of these populations over time. And so by tracking this community over time, we can compare it to changes in the environment over time as well. The survey is still ongoing by Matt Ajamian's lab at Harbour Branch. And this will hopefully provide the framework to expand our knowledge of how elasmobranchs can handle certain levels of change in these highly modified and utilized estuaries. And these types of estuaries, that are utilized by humans and have anthropogenic impacts can be found across the country and across the world. And the IRL isn't special in that regard, but this gives us the opportunity for future studies. And so, for example, considering the survey provides evidence that many of these species do use the IRL throughout their life history, there lies the opportunity to study how anthropogenic sources of toxins and pollutants may affect elasmobranch health, or harmful algal bloom toxins, like I mentioned before. And lastly, the interconnected nature of the related environmental variables that were driving the elasmobranch distribution suggest important implications for future changes in the hydrology of the lagoon. So, for example, if freshwater discharges into the IRL from these flood control canals, if they were to increase in duration and volume, the elasmobranch community could shift even further to dominance by only bull sharks and Atlantic stingrays. And less tolerant species of these low salinities, they may be driven closer to the inlets or even out of the IRL to nearshore ocean habitats. And so any drastic changes in salinity resulting in community shifts would really probably result in decreased elasmobranch diversity, as well as decreased biodiversity of the IRL as a whole. And so in the future, it's likely that human pressures in coastal regions are only going to increase. So knowledge of how species utilize coastal areas will become increasingly more important in order to effectively conserve and manage populations. So we've also submitted our data to the Coastpan program, which will hopefully help refine essential fish habitat designations and nursery designations. So this is a screenshot of a NOAA fisheries story map on the search for Atlantic shark nurseries. And you can see some of our results are outlined here. And so if you want to learn more about this work, in addition to that gear evaluation I mentioned earlier, we do have 2 papers published, which are shown here. And I'd be happy to provide PDFs to anyone who's interested. And then, as I mentioned, the survey is still being continued, and it's going to continue to inform other studies too, which is really exciting. So, for example, because we were able to tag the animals we captured and take other biological samples, we have the capacity for additional studies. So I can mention some here. Tissue samples that were taken during the survey are currently being used by a graduate student in the lab to study the accumulation of harmful algal bloom toxins in bull sharks specifically. Tissue samples were also sent to other colleagues for studies on the genetics of certain species. So for example, fin clip samples have been used in a study by colleagues at the University of Southern Mississippi to show a possible range extension of the Brazilian cow-nose ray. We are currently working on a publication establishing baseline hematological and plasma biochemistry values for certain species. We were also able to acoustically tag several individuals of several different species, and this will provide fine scale habitat use data and information on the movement of these individuals. And then lastly, continuing the survey over time will yield even greater sample sizes and allow for standardized indices of relative abundance to be calculated. And this will hopefully be useful in the stock assessment process for some of these species. I just want to shout out to everyone who helped with this work, including my advisor and my lab. And then, as you can see here, I've listed a ton of names because all the field work wouldn't have been possible without the help of many different colleagues, volunteers, interns. And then I'd like to thank my funding sources, as well as the NOAA Fisheries Apex Predator Program and COSPAN for providing tags, and then all our other permitting agencies. And with that, I'd be happy to take any questions. And if you'd like to learn more about the research, fisheco.org is the lab's website, and you can find out more there as well. And that's it. So Lisa, I'll take any questions.
Excellent. Thank you so much, Grace. That was fascinating. Just a quick reminder to the audience, we're going to take the next 23 minutes, oops, to answer questions. So please continue to enter them in the questions box on the right. And I'd also like to remind you that this seminar is being recorded. If you joined us late or have a colleague you think might enjoy Grace's presentation, please send them to the recording. You can find it on the NOAA Central Library YouTube channel or the Library Seminars webpage later this afternoon. Grace, we do have a couple of questions, so let me go ahead and read the first one to you. Can you describe the salinity of the region? Does the northern region have more exchange through inlets than the southern IRL?
Sure. So the salinity is definitely variable. The range we encountered during sampling was between 0.7 parts per thousand and 42. And the northern IRL, in terms of freshwater, here, I'll go back to my very first slide. And you can see here, sorry, there we go. The northern IRL does start to get more secluded, I guess, especially with the Mosquito Lagoon and Banana River. So as you get into these far northern reaches of the IRL, which I hope you can see with my mouse, there is less exchange with inlets and freshwater. Whereas with the southern IRL, there are more inlets. So yeah, the salinities really varied. It varied depending on where we were, as well as the season, as well as if there were releases from the canals that were happening. But yeah, again, that range was pretty much from 0 to 42.
Excellent. The next question asks, were there many gill net sets where the net had holes that target species may have escaped?
Yeah, that's a great question. There were. We did pull up the gill net quite a few times where there were massive holes through the mesh, which was never fun because I had to repair them. But yeah, so it is possible that target species did leave the net. And so, as I mentioned, we didn't catch mature bull sharks. Yeah, it's very possible that we caught them and they just bust through the net. Not saying that they aren't present in the lagoon. We just didn't catch them. So yeah, it definitely, yeah, it definitely occurred.
Okay, excellent. Next question asks, did you collect any data on non-elasmobranch species caught as bycatch during the survey?
Yes, so we collected data on anything and everything we caught. So our bycatch, the top 2 species were gaff topsail catfish, and oh my gosh, I'm blanking on the other species of catfish. I don't know why, because they're such a nuisance. But yeah, so those were our top 2 bycatch species. But we did catch many other bony fish species. And we recorded how many we caught, and we took length measurements and then released them back. So the data wasn't as extensive. We didn't weigh them, but we did do length and abundance measurements. Sorry, hardhead catfish. Sorry, that's the other one. I was blanking.
The naughty catfish.
Yes.
The next question asks, do you have any recommendations concerning essential fish habitat designations in IRL for the species you studied?
Oh gosh, that's a tough question because I feel like I don't have the authority to make those recommendations, but I would say that our study, as well as other past studies in the other portions of the lagoon, would indicate that this could be essential fish habitat for bull sharks specifically, given that the northern IRL was determined as a nursery for the species, and it's likely that the southern IRL is as well. For other species, I would probably say that more data would be needed, just because we did not catch as quite high numbers of those other species to really be able to elucidate further habitat use patterns and information.
All right. Next question asks, how many tagged sharks and rays were recaptured?
Oh, that's a great question. So we recaught 5 individual bull sharks. Well, sorry, I take that back. We didn't personally recapture all 5 of those, but we got calls from fishermen. So those numbered ID tags, on one side of the tag has the ID number and the other side had our contact information. And so that provides really valuable information because we could see when these fishermen called us saying they caught one of our species, we could see, where they were caught versus where we caught them, and how long of time was elapsed between those 2 captures. And then also, if the fishermen took length measurements, we could get an idea of how much that fish grew in that period of time. So yeah, we had 5 recaptures of bull sharks, and we had some other recaptures of other species as well. I can't recall the specifics, but our recapture rate was less than 1%, which is common in these types of surveys because these species are highly mobile. And like I said, we did acoustically tag some, so that can give us even more information than just the mark recapture part. But yeah, we did have some.
Excellent. This next question actually is also a compliment. Nice presentation, Grace. You didn't catch many bull shark pups in your surveys. I used to catch quite a few further north in the IRL. Any thoughts on why there seems to be fewer pups in your study area?
Sure. That's a great question. We did not catch many. We caught a few. It could be due to the gear. It could be due to, I guess, the different salinities. Really not sure. That's a great question. I'd have to think more about that, and I could definitely get back to that person with a better answer later. But yeah, that is an interesting difference between the northern and the southern IRL.
Yes, just so you know, we don't mention people's names on the presentation, but Grace will have that information and will be able to contact you after the presentation. The next question is, again, another compliment and a question. Grace, this is such a great talk, and the survey clearly captures a great deal of information in animal metrics, tagging, environmental parameters, etc. Are there also other ways to complement the capture survey with other techniques, such as acoustic surveys or camera traps?
Thank you. Yes, so camera traps I think would be difficult in the lagoon, or at least in many parts of the lagoon because of the low water clarity. We have deployed camera gear for totally different studies in the areas closer to the inlets because you can get clear water, especially during the incoming tides from the ocean. But for a really comprehensive camera trap survey, I don't think it would be very feasible because of the low water clarity, which essentially can range from 0 meters to a few meters when you get farther into the lagoon. But acoustic studies, yes, that would be a great complement. And as I mentioned, we did tag some animals, several bull sharks, and then also several spotted eagle rays, and then the smalltooth sawfish were acoustically tagged. And so, yeah, we have all that data. We have acoustic receivers out throughout the lagoon right now, and there's also a network, a telemetry network along the Atlantic coast. So those studies are forthcoming from the lab for sure. It will give us a better understanding of the habitat use and movement of these animals. Yeah, so those hopefully will come out one day. They're still, like I said, still tagging these animals, still putting acoustic tags out. The tags also range in lifespan. So the animals that we tagged, may still be transmitting signals out. So they may still be active. And so the data essentially is still being collected. And so that's why we haven't analyzed any of it yet. But personally, I'm super excited for whenever those studies come out to see where those animals have been going. We know with the sawfish, there were reports, so we caught those down in the very southern part of the lagoon, and there were reports of them farther south on the Atlantic coast, like out in the ocean waters, and then I believe also farther north. So we know that they've been moving around. But again, those tags are still active, so no analysis of the data yet.
Excellent. Another question asks, you mentioned which species had been captured in the previous survey compared to this one. Were you able to make comparisons regarding the abundance of species between the surveys?
Sure. We did not make those comparisons for several reasons. One of the main ones is that we used different gear than the other surveys. So some of those surveys used nets that were larger or smaller mesh. So that will affect if you're going to catch a lot of the little ones or the big guys. Some of those surveys didn't use the long lines. And so, just because we caught, let's say 100 some bull sharks and they didn't, doesn't mean that necessarily there are more bull sharks in the lagoon today than there were before. So because the gear designs between the surveys weren't the same, you can't really make that comparison. And so, yeah, we wouldn't go so far as to say, more elasmobranchs exist now than they did decades ago, or vice versa.
Wonderful. The next question asks, what evidence do you have that the smalltooth sawfish IRL population is recovering?
So I wouldn't say we have definite evidence. I'm sorry if my language wasn't clear, but we just kind of indicated possible recovery because these species were once so abundant and then virtually extinct from the area. And then now we're catching them again, and it's not just us. We caught all different sizes, all different sexes, and in different regions. It's possible. I mean, it's also very possible that these individuals came from, down from the Everglades and came up here, and they're not actually, the population for the lagoon isn't recovered. It's just something that we noticed, and recorded and want to gather more information on in the future. And so we're working with people at FWC. I think their sawfish program, we gave them information. And so it's not, sorry, I wasn't totally clear. It's not, for sure saying the population is recovering. It's just, oh, maybe that's happening. So like, let's keep monitoring. Let's keep track of, what species we're or how many of the species we're catching over time.
This question asks about the gill nets. What were the size of the gill nets in terms of length and mesh size?
Sure. So it was a 200-meter long net, and then the stretch mesh size was 15.2 centimeters, which is 6 inches, and then 20.3 centimeters, which is 8 inches.
The next question is a compliment and a question. Good info. Any thoughts on the value of environmental DNA to complement traditional gear surveys?
Yeah, absolutely. I think there's huge potential there. We did work with some people again at FWC to look at eDNA for possible evidence of smalltooth sawfish in the lagoon. I'm not an expert by any means on eDNA. I know there's tons of caveats, but I think it would be interesting to kind of do a parallel eDNA study while doing the survey and compare the results to see, what are we missing? What species are we not catching that might be there as evidenced by the DNA? So yeah, I think it's a really exciting area of research, but again, I'm no expert, so.
The presentation says differently. So here's another question. Grace, very nice talk. Given that estuarine areas like the IRL are likely to be affected by changing climate, for example, temperature and/or salinity, do you anticipate any specific species could have problems? Maybe this is one possible reason for lack of bulls in the lower IRL?
Sure. I think, yeah, climate change does pose a huge question. Some species do have different thermal tolerances as well as salinity tolerances, as I mentioned. I would imagine that, if salinities increased, got saltier, you would maybe actually find higher diversity because more species can tolerate those higher salinities. And like I said several times during the presentation, if the salinities became lower, I would hypothesize that, bull sharks and Atlantic stingrays, those species that can handle this wide range, especially the lower part of the range of salinities, they would, start to dominate. In terms of temperature, I think you would definitely have some changes. I'm not an expert on the exact thermal tolerance limits of each of these species, but we do know from this study and other studies that certain species were found in still really high temperatures. So, for example, Atlantic stingrays, bull sharks were still found in warmer temperatures. But we did see, I didn't do any, statistical analysis of this, but just anecdotally, higher temperatures, we didn't see as many species. They might have been looking for cooler waters or just not as active, not hunting, or wanting to, feed to be caught in the long line or not moving as much to be caught in the gill net. So yeah, I think it's a definite, climate change could definitely have an impact on the diversity in the community. Yeah, absolutely.
Two more questions. This one asks, thank you for your great presentation, Grace. I was wondering, are there any regular behaviors, for instance, migration movements among seasons for those species? And do you think anthropogenic activities such as sound sources may cause changes in their distributions?
Sure. Yeah, some species do exhibit migrations. So one of the big examples is Knauss stingrays. They tend to migrate south to north, depending on the time of year, all the way from up to the Chesapeake Bay down to the IRL. I will say caveat there, though, we do have colleagues that have studied Knauss specifically, and there might be a certain population that doesn't migrate up to the Chesapeake Bay and instead stays around the IRL. So yeah, there definitely are some seasonal migrations in these species, but I will say, you know, we still caught cow-nose all throughout the year. So the species in general might migrate, but maybe not every single individual. And then I'm trying to think of examples of other species that show migrations. We caught one juvenile black-nosed shark, and that species is also known to migrate north to south from farther south in the IRL around Palm Beach, all the way up to the northern Atlantic. But more information on that would definitely be helpful to get from acoustic telemetry to know exactly when these animals are leaving the lagoon and coming back, because we can't tell that from this study. You know, just because we caught them one day doesn't mean, you know, they hadn't been there for months or they just came into the lagoon. So you really need that telemetry data to show the finer scale movement patterns.
Well, this last question is pretty funny. How many IRL jokes did you make during your master's program?
You know, I actually don't think I made any, which is, I guess, kind of a lost opportunity. But yeah, it's kind of funny anytime I hear the IRL, that abbreviation or acronym. For me, it will always mean the Indian River Lagoon. Yeah.
Well, it's a good in real life experience to have. All right, well, that was our last question. And if anyone has any more questions, you're welcome. We do have like 3 minutes, but meanwhile, thank you so much for sharing your research today, Grace. And Mike, thank you for putting this together for the Knauss Fellows of 2020.
My pleasure.
Absolutely. Any last comments before we conclude today?
Yeah, I would just like to thank everyone who tuned in to listen. I'm really honored and appreciated that you took the time out of your day to listen to this. And again, if you have any questions, please feel free to email me and I'd be happy to talk more about my research or send you the papers that we've published. So yeah, thanks so much.
Absolutely. You have been a wonderful audience, wonderful questions that Grace fielded beautifully. The next 2020 Knauss Fellows seminar will be at noon on Thursday, December 17th. And will feature Connor Fagan, Science and Policy Analyst at the Marine Mammal Commission, as well as Irvin Huang, Ocean Policy Analyst at National Ocean Services Policy and Constituents Affairs Division. NOAA Central Library is very proud to present the work of the NOAA community to support the Knauss Fellows, and I'm truly very grateful to you for joining us today for this library seminar. We hope you will join us again, and everybody, please be well. Thank you all.